Hello everyone, my name is Li Zhou. The topic of my presentation today is iSOM-GSN, an integrative approach for transforming multi-omic data into gene similarity networks via self-organizing maps. Before I start my presentation, I would like to thank my professor, Dr. Roda, for his help and guidance. In this presentation, I will briefly introduce the topic, state the problem, and then describe the proposed methodologies. So now let me introduce two kinds of common cancers, the breast cancer and the prostate cancer. Breast cancer is a type of cancer that starts in the breast. Generally, there are two types of most often breast cancers. The first one belongs to ductal cancer that starts in the ducts that carry milk to the nipple. The second one is lobular cancer. The cancer starts in the glands that makes breast milk. The pathologic stage of cancer takes into consideration the characteristic of the tumor size and the presence of any lymph nodes, metastasis, or distant organ metastasis. There are 5 tumor stages. Stage 0 followed by stage 1 to 4. In stage 0, the cancer cell seems the same as the normal cell. In stage 1, the tumor size is no more than 2 centimeters, or only micrometastasis. In stage 2 and 3, the tumor size grows from 2 centimeters to 5 centimeters, or even bigger. The cancer cells spread to the breast lymph nodes, from axillary lymph nodes to internal mammary lymph nodes to supraclavicular lymph nodes. In stage 4, the cancer cells spread to bone, liver, lung, or brain. The pathologist calls this stage as distant organ metastasis. Prostate cancer is DNA mutation in the cells of prostate. Most often, prostate cancer starts in gland cells of the prostate. This cancer is called adenocarcinoma of the prostate, which makes up 95% of all prostate cancers. The Gleason grading system is most often used to grade this kind of prostate cancer. The Gleason classification system looks at differentiation of cancer cells and the pattern of the cancer cells in the prostate. Differentiation describes how different the cancer cells are. There are 5 patterns of prostate cancer cells based on their differentiation. The lower the pattern number, the more cancer cells look, act, and are arranged like normal cells. Gleason pattern 1 and 2 are not commonly used because these cancer cells look a lot like normal cells. The pathologist will give a grade for each pattern of prostate cancer cells found in the biopsy. The grade of the cancer corresponds to the pattern number. Most prostate cancers have a Gleason pattern of 3, 4, and 5. If the pathologist see only 2 patterns of cancer cells, they will add the grades for each pattern together to get a total Gleason score. For example, if the pathologist gives the cancer cells in the most common pattern a grade of 3, and the second most common pattern a grade of 4, let me write it. Yeah, then the Gleason score will be 3 plus 4 equals 7. If there is a second pattern in the biopsy samples, the grade of the most common pattern of cancer is added to the pattern with the highest grade. This may be the second most common pattern or the least most common pattern. For example, if the pathologist gives the cancer cells in the most common pattern a grade of 3, and the cancer cells in the second most common pattern a grade of 4, and another pattern of cancer cells has a grade of 5, the highest score. So the Gleason score will be 3 plus 5. So it equals 8. To make the Gleason score easier to understand, doctors have developed the grade group GG system. This gives a single score from 1 to 5 based on increasing Gleason scores. For example, the first grade corresponds to a Gleason score 6, and the fifth grade corresponds to a Gleason score 9 or 10. Typically, Gleason scores range from 6 to 10. The higher the Gleason score, the more likely the cancer will grow and spread quickly. Scores of 6 or less describe cancer cells that look similar to normal cells and suggest the cancer is likely to grow slowly. A score of 7 here, there are two cases. So scoring a 7 means that the primary score, larger section of the tumor scored a 3 or 4. Tumors with a primary score of 3 and a secondary score of 4 have a fairly good look, whereas cancers with a primary Gleason score of 4 and a secondary score of 3 are more likely to grow and spread. Scores of 8 or higher describe cancers that are likely to spread more rapidly. These cancers are often referred to as poorly differentiated or higher grade. From this table, we can see that prostate cancer and breast cancer rank first or second in the number of male and female cancers, respectively. And according to statistics, they are still at a high risk value in the 2020 World Cancer List. However, because of the high dimensional characteristics of human genes, it is difficult to establish a predictive model between genes and the disease states using features without spatial correlation. In our research, we try to identify the correlation of genes and their clinical state by integrating 3-omics data, use self-organizing map to do clustering, and use convolutional neural network to do further classification. The datasets are publicly available at cbioportal.org. It contains about 1,000 samples with approximately 60,000 features. In the first step, we made a hybrid feature selection. We filtered features in the gene expression dataset whose variance are below 0.2%, reducing the number of elements from 60,000 to 16,000. Then, we used min-max normalization to normalize data to a range from 0 to 1. Then, we integrate 3-omic dataset based on patient ID and reduce the samples from 499 and 570 to 387 and 392 for prostate cancer and breast cancer respectively. Then we group samples with a balanced distribution based on Gleason score and tumor stage. From these tables, we can see the details on the distribution of the different Gleason groups for prostate cancer and the different tumor groups considered for breast cancer. Finally, we used the multi-CV algorithm to choose the top 14 of the most often mutated genes with a Q value of less than 0.1. Multi-CV is an algorithm to identify genes that are significantly mutated in cancer genomes using a model with mutational covariance. The figure shows on the left a set of chromosomes, each from the tumor of a different cancer patient. Genes are cartooned as colored bands, and somatic mutations are indicated by red triangles. The mutations from all the tumors can be aggregated together by collapsing, and the total number of mutations per gene can be computed. Then the tally is converted to the score and then to a significant level, also called p-values. A threshold is chosen to control for the false discovery rate, also called q-values. And genes exceeding this threshold is reported as significantly mutated. In our case, genes with Q no more than 0.1 were separated as significantly muted. The top 14 muted genes obtained for MUT6CV were considered for the rest of the experiment. The block diagram depicts the main components of iSOM-GSN. First, we create a GSN by extracting features from the gene expression dataset. Second, we use this SOM map as a template and integrate 3-omic data, gene expression dataset, gene methylation and copy number ordination, and construct RGB images. At last, we applied a convolutional neural network to predict disease states of various types. Self-organizing map SOMs, also known as cohonal SOM maps. It is a competitive learning algorithm. The goal of the SOM is to transform the input space into a one-dimensional or two-dimensional discrete map in a topologically ordered fashion. This is an illustration of the chaining of a self-organizing map. The blue blob is the distribution of the chaining data, and a small white disk is the current chaining data point drawn from that distribution. At first, the SOM nodes are arbitrarily positioned in the data space. The node, which is nearest to the chaining data point, is selected. We say it winning neuron. It is moved toward the training data point. Its neighbors, let me change a pen. Its neighbors is highlighted in red. Will also move towards the winning neuron. After many iterations, the grid tends to approximate the data distribution. So in our case, the input is the training samples from gene expression dataset. For each input, it is actually a feature vector and is fully connected to the neurons on the lattice. In our case, the number of neurons is 14 equals to the number of genes we extracted from the gene expression dataset in the hybrid feature selection part. For each connection, it has a small number and we call it weight. At the beginning, we initialize the neurons with random weight, so this is the weight vector. Now, if we want to look for the winning neuron for the GIS gene, for example, this one, we need to sum the Euclidean distance between this gene and the neurons on the lattice and for all the training samples. If the case neuron has the smallest distance to this gene, then we call it winning neuron. We also can call it best match unit. Let's suppose this one. Then this is the winning neuron. Then it is the centroid of the cluster K, and these neurons in the proximity are its neighbors. And then look for the winning neuron for the next gene, and so on. Then we need to adjust the weight of this winning neuron. In this formula, this is the weight of the winning neuron in T iteration. This is the neighborhood function. It is a Gaussian function. This is the learning rate regulation function. Actually, it is an exponential function of iteration. Here, the learning rate is between 0 and 1. In our case, it is 0.05. So the combination of these functions with the red underline, we call it Herbert's rule. And this part, let me change a pen. The combination of this part Actually, it is the weight the winning neuron shares with its neighbors to bring a balance to competition and collaboration. We use the Gaussian function to adjust the neurons in the neighborhood, which are in red circles. dj is the distance between the J's neuron to the winning neuron, and the sigma is the width. Usually, sigma is an exponential function, t is the iterations. So the Gaussian function will shrink over time, and at the end, the winning neuron will not share the weight with its neighbors, because at the end, it will be in the center of the cluster. So the only question remaining is when the iterations will stop. We calculate the distance and identify the winning neuron and update the weights until after e iterations, or when centroids don't change anymore or less than a threshold. That means when they don't move or just jump around, then they will stop. Or when few or no data points change clusters as a result of running the training algorithm, a sum is obtained in which the genes are organized based on their similarity, representing a gene similarity network. This network is represented as a two-dimensional lattice. Now we need to combine three types of data sets as input to feed the template map. Colors can be represented by their red, green, and blue. In our case, red represents the features of expression dataset. Green represents the DNA methylation, and blue is for copy number ordination. And only the features within certain radius of gene in the SOM map template will be filled with RGB colors. Consequently, we will represent the colors as vectors in the unit cube. The parameters of RGB color, red, green, and blue defines the intensity of the color as integer between 0 and 255. If we need more than 3 omic data to integrate with, for example, if we have 4 omic data, we can adjust the RGB parameters. Let it be red, green, yellow, and blue, or any other values between 0 and 255. It depends on you. So now we will have n RGB images. N is the number of training samples. From this sample image, it is clear that it has been filled with RGB values of the 3 omic data. The last step of iSOM-GSN is to feed the RGB images generated to the convolutional neural network to predict the state of the disease. At first, 32 filters of size 3 × 3 pixels are applied to the input in the first convolutional layer, followed by a ReLU function, a max pooling layer, taking the maximum value of 2 × 2 regions with 2 times strides and a local response normalization layer. Then the output of the previous layer is processed by the second convolutional layer containing 32 filters of size 3 × 3 pixels again, followed by a ReLU activation function, a maxi pooling layer, and a local response normalization layer with the same hyperparameters as before. Then, the first fully connected layer receives the output of the second convolutional layer and contains 128 neurons, followed by ReLU and a dropout layer. The second fully connected layer receives the output of the first fully connected layer and output 3 neurons, followed by ReLU and a dropout layer. Finally, the output of the last fully connected layer is fed to sort layer that assigns a probability to each class. The prediction itself is performed by applying a sort max to choose the class with maximal probability for the given test image. To assess the performance, the dataset was split into training and testing with N ratio 70:30. We calculated the main performance metrics that include cataloging accuracy, precision, recall F1 score, and the mean absolute error. In general terms, it is clear that the predicted performance is in the range 94 to 98% with respect to various parameters. So we can say only 14 genes are enough to predict Gleason scores in prostate cancer and stages in breast cancer with very high accuracy. iSOM-GSN shows a lot of potential to apply it to different types of cancer and their respective clinical variables or disease state. Two genes that are known to interact in breast cancer, A1 and GATA3, they appear very close to each other in the iSOM-GSN map. This work was made possible by using the facilities of the Shared Hierarchical Academic Research Computing Network and Compute Canada. There are two limitations. The first one is the unavailability of independent datasets with all data types. The second one is when applied iSOM-GSN on a single omic data at a time, gene expression data alone yielded only 90% accuracy, whereas DNA methylation and the copy number ordination alone yielded only 87% and 89% classification accuracy respectively. This algorithm can be extended to classify pan-cancer data. It can incorporate more than 3 omics for classification. It can be considered as an unsupervised classification algorithm because of the competitive learning nature of SOMs. It also can be applied on other domains. These are the references in my paper. Thanks for your time, and feel free to ask me any question about this paper.